This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 158, recorded on August 3rd, 2017. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today, from Small Things Considered, Elio Schaechter.
Well, hello there. How are you?
I'm well. We're in August.
Yeah.
The summer is going by.
Yes.
You know what's interesting? Today, the new medical students are moving in.
Oh, boy.
August 3rd. Wow. All the streets are blocked with U-Haul trucks. And, of course, that voice you just heard is from Ann Arbor, Michigan, Michele Swanson.
Hello.
Are you in August as well?
Oh, yeah. But the nights are cooling down. You know, it gets into the 60s at night, so It's good sleeping weather.
And joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
I just got back from Montana.
Ooh.
I was at a meeting at the Rocky Mountain Laboratories.
What a great place.
You were there last year, weren't you?
Yeah, I was there in November, yeah.
Oh, November, yeah.
I like it. I like it there. It's very dry, so during the day, it's 35 Celsius, almost 100 degrees, and then at night, it goes down to 15 Celsius.
Ooh, sleeping weather.
But
no air conditioning.
Actually, a lot of people don't have air conditioners. They say if you have trees around your house, you don't need air conditioning. But it's really pretty there. And I have to tell you something. My colleague Dix, so we were there to do a TWiV at a virus meeting, and I brought my colleague Dixon Despommier with me because he's a big fisherman and he loves to fish there.
Ooh, trout.
So he's a fly fisherman, of course. So he said, you have to come with me. So I spent a day floating down the Bitterroot River with him. It was really cool. I didn't fish. I actually just videotaped him fishing, but he's a really good fisherman. He must have caught 20 different trout. But what I found cool was the whole ecology of the whole thing. You know, this river is going through a pine forest, and in the pine forest live these little white moths, and occasionally they get lost and they come out and they fall into the river, and then trout eat them.
Wow.
And you can see the trout eating them, and then that's where you cast, and your lures look like these moths. All of this is new to me, and I'm sure a lot of listeners know all about this, but I just found it really cool.
Wow. So is Hamilton working its way up on your list of destinations?
No,
Another residence?
I think it's probably a bit remote for me to live there.
That's the good news and bad news, right?
I think I need something more on the order of Chapel Hill, Ann Arbor. You know, Ann Arbor would work for me. I don't think my wife would want to move to because it's too cold in the winter. She wants something warmer.
It's getting warmer every year.
Global warming.
Well, actually, in Hamilton, there were all kinds of forest fires burning. The skies were full of smoke because it's getting hotter and hotter and drier. It's quite clear. And, you know, one of the scientists at Rocky Mountain Labs, his house was burned down last year because these forest fires come over the mountains into the valley and then burns up everything.
Who was that? Who is that, Vincent?
Byron Coy. He works on prions.
Okay.
All right. Today we have two very interesting papers for you.
I'll say.
And the first one is a snippet that I asked Michael to present, as I found this very recently, and I thought it would be interesting. What'd you think of this, Michael?
It was a very cool paper. I very much enjoyed reading it and learning about it, and it continues to fascinate me as to how the microbial world is everywhere. As many of our listeners know, the most popular undergraduate major today is neuroscience. And this is a blend of neuroscience and microbiology. And the title of the paper is A Prokaryotic Viral Sequence is Expressed and Conserved in Mammalian Brains. And it was published in the Proceedings of the National Academy of Sciences, or PNAS, and it was authored by Yangwei Ye, Vignesh Gunasakarina, and Laura Manuelides. And they're all at the section of neuropathology at the Yale University Medical School. And, this was a bit of a reminiscent. And they start their paper by telling us a natural and permanent transfer of prokaryotic viral DNA. So these are phage sequences to mammals has not been reported by others. So this is big news unto itself. And some of you may recall from our very first episode of TWiM, when we not only introduced you to this notion of discussing science on a podcast, but the concept of how horizontal gene transfer from a human host to a bacterial pathogen could occur. You might recall that that paper was about these long interspersed elements, which was from a paper where the authors reported on the identification and characterization of horizontally transferred fragments of the human long interspersed nuclear element L1, hence the term lines, to the genome of the strictly human pathogen Neisseria gonorrhea. And we didn't quite understand what to make of, human DNA going to a bacterium. And our discussion of lines is back to some extent, as our understanding of them is partially required to consider the significance of the snippet that I'm going to present. And so numerous copies of these lines or line elements were initially isolated from human and mouse nuclear DNA, and they were shown to be homologous with each other and organized together into this megabase GIMSA band domains on our chromosome arts. And so lines, we still really, our understanding of them so remains incomplete, but Mother Nature never carries extra luggage. She always is getting rid of things that she need not carry forward to the next generation. So our knowledge of these are incomplete, but today's snippet takes us the other way, where we'll learn how the introduction of prokaryotic viral sequences, albeit a long time ago, were moved into the mammalian brains with their subsequent vertical or germline transmission up our genetic lineage, and in a manner where the data from this paper implicate maternal cytoplasmic inheritance, as is used for the propagation of the protected mitochondrial DNA. And that may or may not be responsible for, and where these authors, the neurosciences, are going with this, is they're trying to understand whether or not this viral sequence from a bacteriophage may be responsible for some of the horrific pathology that is associated with these infectious proteins known as prions. And so as we begin to tear into the meat of this paper, not only are we going to learn that this prokaryotic viral DNA has gotten in, but it's actually being expressed in mammalian brains. And that this DNA, when it's found, is actually protected. It's nuclease resistant. And more importantly, the protein it makes is also resistant to proteinase K digestion. So here's the story. And I don't know if they named it or these.
It sounds a little bit like a prion, but it doesn't have anything to do with prions directly, does it?
No, it doesn't, or does. You sort of, as you're reading the paper, you're trying to make that connection, but then their data show you that there is no connection.
So I should say this group studies prion diseases.
Mm-hmm.
And I think they initially thought, so I have to say that everyone pretty much has accepted that prions are protein-based infectious agents, right? This group contends that there's a nucleic acid associated with prions and prion.
They keep wanting to put it in.
And so they've published papers. In fact, there's one in the reference section. The title is Scrapie and Creutzfeldt-Jakob Require Agent-Associated Nucleic Acids. That's a 2016 paper. And of course, I personally feel, and many others do, that there is plenty of evidence indicating that prions don't need any nucleic acid except the genome that encodes them, of course. But the actual agent itself is just a pure protein. So it's a little weird that this group still feels that. But I think their initial investigation into this particular DNA was to try and see if it was one of these prion-associated DNAs. But it turns out it's also in uninfected cells. So that's certainly not the case, you know.
And the name of this DNA, it's referred to as Sphinx DNA. And this Sphinx DNA,
That's a good name.
the Sphinx DNA is less than 5 kilobases in size, and they were previously isolated from nuclease-protected cytoplasmic particles from rodent neuronal cell lines and brains of various mammals. So you can now begin to understand, after Vincent's comments, why these folks were beginning to go down this path of looking at this. And 2 of these DNAs were sequenced after Phe29 polymerase amplification, and Phe29 is a bacteriophage, correct, Vincent?
That's right. Yeah.
Yeah, it's a bacteriophage, and it, upon sequencing and looking at the sequence, it revealed significant but imperfect homology to segments of the commensal Acetobacter phage. Now, Acetobacter, as you well know, as we have discussed in the past, is this gram-negative gamma proteobacterium that is in the Pseudomonales order, and it's a pretty nasty microbe. It's an environmental creature that literally acquires antibiotic resistance traits just by looking at a gene. It effectively becomes resistant to it. So it's a pretty horrific pathogen in the clinic, and yet it's an environmental creature that normally doesn't, it's not fermentative, so we don't associate it with our guts per se. It's more of an environmental passenger that comes in. So it's really unusual that it's involved here. The other remarkable thing is we need to fundamentally ask, how did phage DNA get in? Since normally the brain is isolated from microbes. Though you may also recall 100 episodes ago on TWiM 58, we talked about the brain microbiome.
That's right.
So
that's right. And viruses get in the brain all the time, right?
All the time. So
I'm not sure how isolated the brain is.
Yeah, but
you've got viruses on your brain.
I do,
Vincent.
and on my license plate as well.
Yes. So
What does it say?
My license plate says viruses.
Hey. Okay.
So the other piece of background information that you need as you read this paper is that this Sphinx DNA contains some genetic element called an iteron, ITERON. And these things are directly repeated DNA sequences which play an important role in the regulation of plasmid copy number in bacterial cells. It's one of among the 3 negative regulatory elements found in plasmid. And the rule of thumb of remembering what negative and positive regulatory elements mean is something has to be taken away from the regulatory element in order to get the expression from that element. That's negative. Something has to be taken away. And a positive regulatory element is something has to be added to the genetic element in order to get it to go. And the others that are included in this class are antisense RNAs, which many of you are well familiar with. And then something else that you may not have heard of before is these counter-transcribed or ctRNAs, which is a plasmid encoded non-coding RNA that binds to the messenger RNA of the replication initiation protein B, which then results in translational inhibition. Remember, in prokaryotes, transcription and translation are coupled. And so these iterons are in there with it. And the iteron, if you will, these directly repeated DNA sequences, if you look back at what a line looks like, you can begin to see some similarities. So what they've done in this paper is they've asked the question, did this 1.6 kilobase DNA sequence that they named Sphinx 1.8 with its interon before its open reading frame get expressed in neural cells and brains? And when they specifically looked at specific neurological pathologies of when they add prion proteins and they add the infectious form of the prion protein. Now, here's where the authors got really lucky because they're dealing with a prokaryotic viral protein. And so the tools that they had at their disposal is they could synthesize the protein and they could synthesize pieces of the protein. And what they ended up doing is they made rabbit affinity purified antibodies generated against a peptide that had no homology to a mammalian sequence, and it was non-glycosylated like all good prokaryotic proteins are. And it made it available to them so that they could look at, brain chemistry or brain tissue or neuronal tissue or
localization.
And they could
localization above all.
And they could figure out where this stuff was. And so the other thing that they got lucky on is that this Sphinx protein is resistant to limited proteinase K digestion in Triton X 100. And this then enables them to compare and contrast it to this whole prion-dependent pathology, because PRP, which is the non-pathogenic form, if you will, of the host prion protein, will effectively be dissolved by proteinase K in Triton, while the amyloid variant, which is the one that gives you the shakes and all the other bad things associated with prion disease, is resistant to proteinase K and Triton X. So you got to keep those two little factoids in your head as we go through it. So not only did these authors learn that the Sphinx 1 was expressed in brain, but its activity was concentrated in selected brain synapses. It goes to what Elio just tumbled to, namely the localization. And it was specifically being associated with the anterior motor horn neurons. And sitting in neuroscience seminars here at the Medical University, I've learned from attending seminars is that particular thing serves to integrate many complex neural inputs. And that's the extent of my knowledge of what the anterior motor horn neuron does. I need to go back and take undergrad neuroscience, I think.
But the synapse is the business end of the
absolutely
neuron, I would say. Mm-hmm.
Yes.
That's that's where the information is passed from one nerve to another, right?
Mm-hmm.
Correct.
To allow the transmission.
Yep.
So in the meat of their experiments, they proposed that commensal and or environmental viruses might initiate or be involved in the late onset of these neurodegenerative diseases. And so these neurodegenerative diseases, they coined the term, are transmissible encephalopathies, and they abbreviate this throughout the paper as TSEs. And then, of course, that we've already introduced you to the term Creutzfeldt-Jakob. It's also known as Kuru, which was a disease of Aboriginal people in New Guinea, where they would eat the dead male elders, and they would transfer this infectious protein to the young people. And of course, in England, they of course had scrapie, where they would feed ground up brains from cows to sheep, or the ground up sheep brains, and they would transmit.
So scrapie is a sheep disease.
Correct.
Naturally occurring. What they did in the UK is they transmitted it to cows by feeding cows ground up sheep. And that created a new TSE called mad cow disease, or bovine spongiform encephalopathy. And then people would eat the cows, you would eat meat, and you cannot inactivate prions by any cooking. And there was an outbreak of BSE transmitted to people for a number of years in the UK.
And of course, anyone who works in a surgical suite knows that if anybody has any symptoms of CJD, you of course have to document all the instruments that were used in surgery, and there's special procedures for cleaning those instruments subsequent. And there's no good bioassay to determine if they're sterile. And it's really a very complicated
However, this paper deals with something that's connected to prions. It's not about prions.
Right.
No. It's these
So this sequence, the thing, and also the staining, the production of the protein, as you say, detected by the antibody, is also in normal tissue, not just disease tissue.
And that's absolutely correct. And that's where their paper sort of fell apart from their central hypothesis. Yeah. They really set the paper up beautifully. They had a bunch of cell lines. They had infected and control animal groups. And the unfortunate thing is, again, they have these very careful and categorized antibodies selected against these open reading frames that bore no homology to mouse or human or any other known mammalian nuclear or cytoplasmic sequence in the database. And they asked, was the Sphinx 1 protein differentially expressed in this TSE agent infective brains? And so they did a very straightforward experiment that many first-year graduate students do, namely Western blots. And they analyzed both the proteins in brains of wild-type mice, TGA20 mice. The mice go on, and in copies with PRP. Recall that PRP is sensitive to proteinase K digestion in Triton X, and PRP null mice, and then, of course, ones with the amyloid protein. So this is the twist on their experiment. So they asked on the blots, looking at the digested proteins from the suite of animals or cell lines that they were looking at, whether or not it was behaving like the neurodegenerative phenotype or the control. And unfortunately, it was behaving the same across the experimental and control lines. And so under these standard conditions, every control and infected brain showed the same amounts of proteinase K resistant full length SPX1 or Sphinx 1 equal to about 8% of undigested samples.
Michael, is there any sample that did not have SPX protein in it? Do you recall?
No, I don't recall, but I don't think it did because they use the word all.
So this is weird because it's both human and mouse that they look at. It's everywhere. I don't understand why. Unless it's really ancient, right?
Well, they found Sphinx in everything that they've looked at, and that was in their introduction. They found Sphinx in cow's milk. They find it in brains. Everywhere they look, they're beginning to find this sequence. But the really neat thing is when they began to do some really fine structure histology, the last figure of their paper. In the brain, the protein, the Sphinx 1 protein, concentrated in the terminal synaptic bouton. And recall that this bouton is the enlarged part of the nerve fiber. When everybody probably has an image of what a nerve fiber it looks like, and it's this bulbous end. And that's where this protein is concentrating, where the axon forms the synapse with the other nerve. And this bulbous end is the business end. So the bottom line question I guess they were originally trying to ask was the Sphinx 1 responsible for CJD? And the answer, unfortunately, was no, because the isolated neuronal cell lines in normal brain produce SPX1, and there were other latent circular DNA elements. So this is where Elio normally chimes in and says, stay tuned for more, because they found other sequences from bacteria, and including a 2.4 kilobase DNA. And they end their manuscript by saying that needs to be further evaluated to see how well it's conserved in mammalian brains and other differentiated tissues. But the authors have provided us a roadmap of how to dissect this complex, if you will, prokaryotic neurodegenerative disease axis. And we're not any closer to figuring out what Sphinx 1 does, but like real estate location matters. And here their detailed tissue-specific characterization really, because they made such good antibodies, they have the reagents, they help us really appreciate how to do the science. So summing up the paper, you have the Sphinx 1 protein that's nuclease and protease protected. It's a viral element that originally came from the environment. There's surmise that likely invaded via wounds or were taken up by cells in the intestinal tract after the bacterial hosts were digested by pancreatic enzymes. And these viral DNAs probably persist and spread in animals as symbiotic elements that developed into functional and essential mammalian collaborators during evolution. So we really don't know what this Sphinx element is doing, and it may be the upstream interon that is responsible for this. It may be a way of giving that nucleic acid or the surrounding nucleic acid around it something special in terms of evolution. And because Sphinx DNAs were initially isolated from these cytoplasmic particles that were resistant to nucleases, they probably reside and replicate in the cytoplasm. although the authors maintained they were not able to formally rule out nuclear episomal resonance. So it's a fascinating story. It's the first of the kind where we begin to see prokaryotic viral sequences go into something remarkable like the developing brain. So it was a fun paper, Vincent.
My conclusion, if I may,
you may,
is that the people who are going to go into neurosciences better take a microbiology course.
Well, they need to know what a phage is. They need to
Absolutely.
They need to understand what the interon was, which is why I went into describing what it did, and more importantly, what it did with its antisense RNA as well as that ctRNA, which I don't think many folks appreciate. This plasmid-encoded non-coding RNA that actually binds to messenger RNA, RNA controlling RNA, which ultimately causes translational inhibition. And so it's really giving us a peek, if you will, at the second genetic code that we all have to contend with.
So, Michael, is it fair to say that they tested Koch's postulates and they were not fulfilled?
I think that is a fair statement, Michele.
So the cool, scary biology of all these prion-type diseases, there's no evidence yet that these are contributing.
Not
right?
The Sphinx 1. Not the Sphinx 1.
I don't know how this would replicate in the cytoplasm. It's too small to encode any polymerase of any kind. And, DNA, as far as we know, in eukaryotic cells, DNA doesn't, except in the mitochondria, DNA does not replicate in the cytoplasm. So it's a little weird. I don't know why all these different species have the same kind of sphinx in them. That implies it would be really old. Mm-hmm. But, we know that the mitochondrion invaded eukaryotes or archaea many years ago, and we got a lot of genes from it. But why is this old? I think there are lots of questions here. It's kind of provocative, but on the other hand, I'm worried about a lot of aspects of it, especially finding it in so many places.
Yeah, and they talk about how old it is by comparing it to a modern day Acetobacter phage, mm-hmm. and it's sufficiently different. So you can go and do the molecular chronometry, but unfortunately, there's no ribosome. So your database is only as good as what your watch is measuring.
Anyway, I thought it was interesting. We'll see what happens. The real key is whether others duplicated and extended and so forth, or whether this remains an isolated observation. We'll see. Thank you, Michael.
Thank you.
All right, our paper today is all about diarrhea.
Oh, my.
Paper published in Cell Host and Microbe, and the title is IL-22 upregulates epithelial claudin 2 to drive diarrhea and enteric pathogen clearance. And there are a whole host of authors here. We have two co-first authors, Pei-Yung Tsai and Bingkung Zhang. And then the senior author is Gerald Turner. They're from China Agricultural University, University of Chicago, Soochow University, Osaka University, Harvard Medical School, and the University of Texas Southwestern in Dallas. And this paper is addressing the question, is diarrhea good for you? Does diarrhea clear enteric pathogens? This is a question that has been debated for years, and we really don't have an answer, and they try and provide some answers here. So when your intestinal tract is infected by a microbe, often, not always, but often the integrity of the intestinal barrier is compromised. Of course, the intestine needs to keep fluids inside the cells. When that's compromised, you have diarrhea. And some microbes can damage the epithelium. Invasive and toxogenic organisms can damage the epithelium, and that, of course, breaks the barrier and fluid comes out. But there are also non-invasive microbes, which we call attaching and effacing pathogens, that inject proteins into the eukaryotic host cell by type 3 secretion systems. And those proteins that they inject lead to increased intestinal permeability and the loss of fluids. But the real question is, what is the role of this in pathogenesis? So they use a mouse pathogen, Acetobacter rodentium, and this is an attaching and effacing enteric bacterium that causes colitis. You feed it to mice, they develop colitis. So before we get into the data, let's talk a little bit about the barrier in the gut tract. Of course, the gut tract is lined with epithelial cells, and these are joined by what we call tight junctions. The tight junction is a complex of protein that bridges two epithelial cells, and it's the barrier that keeps things from leaking out. Of course, if you break the cell itself, fluids will leak out, but there are fluids beneath the cells, the subepithelial space, that are kept there by this tight junction. And there are three ways that transport occurs across this tight junction. First is what they call the unrestricted pathway. This lets everything out, basically, and everything
chaos,
in. Chaos,
damage, chaos.
I'm trying to think of some human analogy. Ions, water, macromolecules, bacteria, viruses. This is when you damage the cell, you damage the tight junction, and everything is out of control.
It's like bursting a balloon.
Bursting a balloon, everything comes out. Then there's what's called a pore pathway, and this will let out things that are 6 angstroms in size. So this is charge and size selector. And then there's what's called the low conductance leak pathway, which will let things in and out up to 100 angstroms. Okay, so those are 3 different kinds of transport. And we're going to assay them individually in this paper. And in fact, that's what they have done here. They've developed a cool assay where they can measure the contribution of the unrestricted pathway, the pore pathway, and the low conductance pathway. And
let me just interject. They did a beautiful job. The very first panel is a lovely schematic that lays out the biology and also the reagents that they use for these 3 different pathways, the pore, the leak, and the unrestricted. So it's a great aid to following the paper.
So the idea is they feed mice the Acetobacter, and then they feed them the 3 molecules of different sizes that can distinguish between these 3 different transport pathways, and they all can be measured relatively easily. And the molecules are creatinine, fluorescently labeled 4 kilodalton dextran, and rhodamine labeled 70 kilodalton dextran. So in other words, fluorescent, a green-colored 4 kilodalton dextran, and a red-colored bigger one. And these are for...
Creatinine is about what? What's about 500?
Yeah, so that's for the 6 angstrom pathway. That's small.
Right.
The 4 kilodalton dextran is about 28 angstroms, and so that does the intermediate one. And then the other one is 120, which is the unrestricted pathway. And
which is the creatine. No, the creatine is the small
creatine
one.
is a small one. The 4 kilodalton dextran is the intermediate one with the up to 100 angstroms, and then the 120 angstrom, the 70 kilodalton red dextran, you get the unrestricted pathway.
Boy.
And then what you can do is measure these in serum. So if they can get into the epithelial cell, then you will find them in serum, and they're easy assays. Of course, the red and green are really easy to pick up, and the creatinine, you have an assay for that as well. So that's the assay. You feed the mice, you measure these. So basically what they find is that by 2 to 3 days after you infect mice with Acetobacter, you can see the pore and the leak pathways activated. So the smaller molecules are getting into the blood. And then by 6 days, you see the unrestricted pathway. So early on in infection, small things can be getting in and out, presumably, right? Because they're measuring things getting in, but things can also get out as well. And then by 6 days, you have enough damage so that you have unrestricted diffusion. Okay, so how is this happening? They focus on this tight junction protein, which is in the title of the paper, called Claudin-2. And it's known from previous work that...
Why don't you spell it out?
Claudin, Claudin, and -2. There are a number of different claudins, but this one is known to regulate intestinal permeability by the pore pathway. All right, the pore pathway, just to remind you, is the small one, 6 angstroms. And in fact, what Claudin-2 does, it makes a pore in the tight junction that's small, and it allows sodium and water to pass through it. Those are pretty small molecules, right? And so Claudin, if you overproduce Claudin, it will make a pore, and it will allow sodium and water to pass through. And they see in mice, they look in infected mice, and you can see that Claudin-2 is specifically upregulated by 2 days after infection. And they look at RNA, they look at proteins, they look at immunohistochemistry to see in the intestinal epithelium, and they can see that it's upregulated by Citrobacter infection. At the same time that they see high Claudin-2, they also see an increase of a cytokine called IL-22, which suggests a connection between the two of them. And in fact, they do a series of nice experiments using intestinal, what we call epithelial organoid cultures. These are cell cultures that differentiate to look like the structure of the epithelium in the intestine. They can show that adding IL-22 to these turns on the synthesis of Claudin-2. If they inject IL-22 into mice, Claudin-2 goes up. And finally, they have mice lacking the IL-22 gene. And if you infect those with Citrobacter, there's no increase in Claudin-2 at 2 days after infection. So they've done
just to keep in mind what Claudin means. I just looked it up.
Mm-hmm.
They were named in 1998 by two Japanese, Furuse and Tsukita, and it comes from the Latin word claudere, which is to close.
Ah,
which is very appropriate.
Beautiful.
Huh. Of course, if you make a lot of Claudin, you open.
Right.
You open the
That's right.
small ball. I don't know why it's the opposite. Anyway,
yeah, you're right.
So much.
Well, maybe that's because it's 2.
It could be that one of the others
Claudin
closes.
1
Maybe, yeah.
Yeah.
So this is a nice part of the paper. This is the first time this has been shown. This IL-22, which would be, a cytokine would be something produced by infection, could be a bacterium or a virus, and it turns up Claudin-2. And that is going to be important. They also have mice lacking the Claudin-2 gene, and they also have mice that are transgenic for Claudin-2, so they make more of it than normal mice.
And it's not a lethal mutation.
It's not lethal, although, that's interesting that Claudin-2 is not lethal. Absolutely, yeah. So they can do these experiments. The IL-22 deficient mice, they have to look at early in infection because they have problems controlling the infection. But this Claudin-2 knockout and transgenic are really the key reagents in this paper. Now, if you just take a transgenic Claudin-2 mouse, so now they're making more Claudin-2 than a normal mouse would be. If you just take uninfected transgenic Claudin-2 mice, they have more sodium and more water in their feces than control mice do. Okay? And that makes sense because, as I said before, if you make more Claudin-2, it makes a pore in the tight junction and it lets out sodium and water. There are mice that don't have any Claudin-2, have the same amount of sodium and water in the feces as wild-type mice. Okay, so now they infect these two different mice with Citrobacter. Wild-type mice, when you infect them with Citrobacter, they develop a colitis, but they don't lose any weight. They don't get very sick. And this is the same for the Claudin-2 transgenic mice. However, the Claudin-2 null mice that can't make Claudin-2, if you infect them with Citrobacter, they have significant weight loss. They have a lot of Citrobacter in their feces compared to wild-type in transgenic mice. And if you look in the mucosa of these animals, you can see the bacteria have penetrated into the mucosa. Now, normally in wild type and the Claudin-2 transgenic mice, the bacteria stay on the surface of the epithelial cells, and they don't penetrate into the epithelium. They don't cause extensive epithelial damage. But if you take Claudin-2 away, then that's what happens.
And their data for this is just exquisite, including beautiful pathology where different components are labeled different colors. So in an instant, you can get from a photograph the biology. It's really a striking difference between the wild type and the Claudin-2 knockout, where you see the bacteria are winning. They're just taking over.
And here's the key. When you take away, in wild type and Claudin-2 transgenic mice, you don't have a lot of mucosal associated bacteria. In fact, there are far more mucosal associated bacteria in the Claudin-2 mice. So the implication is that Claudin-2, which we've already shown allows water and sodium to leak out, if you take that away, you have more bacteria sticking to the surface and penetrating the surface of the epithelium.
So it's starting to sound like diarrhea is good for us.
And at least in mice, right? Yeah. Infected with Citrobacter.
What do you suppose all these other Claudins do? There's up to Claudin 15 here. There's no hint as to what they do.
Well, are they all involved in the tight junction? Do you know?
But not this way. They're not involved in
this pore formation. Yeah, I know Claudin-1 is part of the tight junction, and that in the liver helps hepatitis C virus to get in. It's part of the receptor for hepatitis C virus. Of course, that's not its real role, but it's one thing that we know it does. So as Michele said, it's starting to look like Claudin-2 diarrhea may be good for you. So let's explore this a little more. Claudin-2, they also see that when you take Claudin-2 away and you infect mice, you get many more what we call pro-inflammatory cytokines in mice lacking Claudin-2. And so these are produced, of course, upon infection, and it looks like Claudin-2 limits this immune activation. Whenever you have an immune response against a pathogen, you want to limit it because otherwise you have pathology caused by the immune response itself. It looks like Claudin-2 seems to be involved in that. Now, they further look at the epithelium of these knockout mice. They look at proliferation of the epithelial cells in the gut, the cell death, apoptosis, and histopathology. Infection causes all of this in wild-type mice, but in knockout mice, in the Claudin-2 knockout, it's much greater. So in other words, if you take away Claudin, you get diarrhea, and you get much more pathology and cell death in the intestine. Now, what's the bottom line here is that fecal water in knockout mice, Claudin in knockout mice, seems to be secondary to intestinal damage. Okay, so the idea is that early in infection, Claudin-2 is upregulated. It's a host defense protein, basically. It's upregulated, and it allows water to flow out, and that washes the bacteria away, which prevents subsequent damage. So,
in the solution to pollution is dilution.
That's right. Remember, Claudin-2 is a pore. It lets out water and sodium. Now, to test this hypothesis, they say, let's do something that doesn't involve a Claudin knockout mice. So what they do is they induce diarrhea artificially with something that many of us are familiar with.
Of a certain age. Those of us with a certain age.
Polyethylene glycol, which pulls, if you ingest polyethylene glycol, it pulls water out of your intestinal mucosa and you get diarrhea.
Called Miralax.
And by the bucket load.
And if you have gone for a colonoscopy to cleanse your colon beforehand so that they can see things, otherwise it's covered with fecal material, you take polyethylene glycol and you have
a liter of it.
A liter. You have a rocky night.
You have a very bad night, yes. And you can only drink fluids, clear fluids. And it's horrible. It's worse than the actual procedure, but we don't need to describe that. So they treat them.
That procedure is important for you.
Of course.
It is.
The procedure is important. We don't want to get letters from the colonoscopist.
No,
for clean as a whistle.
It's as clean as a whistle. In fact, once I was awake and I saw the video monitor, and what a clean colon it was, pink and lovely looking before I passed out.
Cleanest colon in New York City.
But these mice now are fed polyethylene glycol. And if you do that in infected mice, it reduces the number of Acetobacter that attach to the mucosa. It kind of makes sense, right? You induce diarrhea, you wash away, as Michael said, dilution. Solution is dilution. Now, if you feed transgenic Claudin mice PEG, there's no effect because they already have water efflux increased without PEG, right? Because they have increased Claudin-2 and they wash away their Acetobacter. There's no effect of PEG on Claudin expression, so that's not why PEG is doing this. PEG treatment prevents colonization by Acetobacter. It causes decrease in the inflammatory cytokines. It reduces proliferation of the epithelium. It reduces histopathology. It reduces apoptosis. So polyethylene glycol promotes the exit of water. It promotes the clearance of bacteria very much as overexpression of Claudin-2 does. And so, they've end up their paper by looking at their probes again to see which transport pathways are being altered, and they find that polyethylene glycol decreases the unrestricted pathway, and that's the one caused by epithelial damage. So in other words, by moving out the Acetobacter, you don't get epithelial damage, which would cause unrestricted flow of their big dye in and out of the intestine. So PEG pulls out water. There's no pores or no damage involved. So that's the bottom line. Early in infection with this particular intestinal organism, Acetobacter, infection upregulates Claudin-2. It's kind of an immune response to infection. That causes the formation of a pore in the tight junction. Water comes out, the mice get diarrhea, flushes away the bacteria to prevent damage that would happen later on by 6 or 7 days. If you didn't have an upregulation of Claudin 2, like the mice that don't have Claudin 2, you would have replication, damage to the epithelium, and even more severe diarrhea later. And PEG corrects the defect and knockout mice that don't have Claudin because it pulls out water as well. So the bottom line is that for these mice infected with Citrobacter, diarrhea is good. And that's the kind driven by these little Claudin 2 pores or by PEG, not by epithelial damage. And so they say that maybe, can we extend this to people? Maybe early in infection. So if you sense, the early days of having diarrhea, if you've eaten something and suddenly you're getting diarrhea, you should probably not take something that suppresses the diarrhea.
Mm-hmm.
Because if you do, you're going to suppress the clearance of bacteria, and that will lead to more severe disease later. That's a suggestion. I don't suggest that anyone try that because we don't know what goes on in people, right?
But I think it's time.
At least in cholera, this doesn't quite pertain because the cholera bacilli stick to the wall very nicely.
That's right.
But that's a toxin.
You don't get rid of them by the diarrhea doesn't get rid of them.
So that's a
toxin-mediated disease, and the toxin is responsible for the hypersecretion.
Yeah, so this would be only for attaching and effacing bacteria that inject things by type 3 secretion would this apply, right? Because those are not going to be causing the epithelial damage.
Which in people is mostly known as traveler's diarrhea, the Montezuma's revenge when we go to Mexico. And similarly, they call it Yankee revenge when the Mexicans come to the U.S. and get the same thing. The E. coli that you haven't seen before effectively responsible for this.
Now, let's say they did a clinical trial and they compared people with and without anti-diarrhea treatment, during an episode. And it turned out that, it is better to let the diarrhea run its course. I don't think a lot of people would listen because, if you're traveling and you want to do things and you get traveler's diarrhea, it's a problem because you end up going to the bathroom quite often, right? And people... You
spend the night adoring the porcelain goddess.
Yeah, but it's self-limiting and you avoid doing tissue damage, right?
You avoid the tissue damage.
That would be the idea.
Amp up the immune response.
Yeah.
So I think this paper is going to go a long way toward starting to reduce the stigma associated with diarrhea. Next time you have diarrhea, don't be ashamed. You can be loud and proud. It's part of your immune system.
Oh, no, that's the title of the show, Michele. Loud and proud. You've now done it, Michele.
That Vincent began his summary by saying the bottom line.
That's another.
Oh God, we're gonna get letters for sure.
That's another title, the bottom line.
But at least we didn't say it fills the cup.
That's great. I like that. Yeah, I notice on Twitter a number of people wearing buttons. People who work on
diarrhea,
and they say, "Ask me about my copious diarrhea" or something.
But thinking about the medical students moving in at Columbia, we introduce them to diarrhea, but I think they would appreciate the basic science and understand the immune reaction if they just took a look at this paper and actually sat back and marveled at how remarkable the human condition and this balance between the microbial world that we have. And they would really understand the importance of tight junctions and pore size, and because they're going to do that as year one medical students. They're going to learn about the healthy system.
Cytokines that recognize the infection and amp up the pores so we can get the outpouring of fluid. It's really beautiful. And I want to add that the paper is very well written. They lay out the rationale, they explain their assays very clearly, and each of their figures, they do everything they can graphically to lead your eye through the different components of the data. It's really well done. It would be great for anybody looking for a paper for Journal Club, for example. Plus, you get to make lots of diarrhea jokes. I was able to correspond with the two co-first authors. So if I may, Pei-Yin Tsai is got interested in physics even as a high school student. She was interested in electromagnetics, quantum physics, optics. So she majored in physics in college. But in her junior year, her mentor encouraged her to broaden her interests a bit and consider a different field. So she was originally doubtful about the wisdom because physicists use mathematical models to predict and describe phenomenon, wave propagation, gravity, etc. She really had limited knowledge of how biologists and immunologists do their research, but she was emboldened by her mentor's encouragement and her own curiosity. So she took some molecular biology and cell biology classes, and they really opened her eyes. She realized that her training in physics actually helped her excel in biology because she perceived things in a different way, and plus she learned that she liked it. So she decided to continue on then and got a master's degree at Taiwan University in immunology, where she graduated first in her class, surprising some of her classmates because they knew she was a physics major. But she credits her success to a strong desire to increase her knowledge and also realizing that her skill sets in physics and mathematics were an asset in immunology because many of these cell mechanisms are really complicated and they can't be easily explained by mathematical models. So
Amen.
she then, I remember you saying that in our last episode, Elio. So then she decided to continue on. She got a PhD in immunology at the Max Planck Institute in Dresden, studying microRNAs and regulatory T cells. She then went to the University of Chicago for her postdoc, and that's where she started studying this Claudin-2 tight junction protein. And she said she always had wanted to shift to a mouse model, and Professor Turner, her advisor, supported her and gave her a great project and resources. She says that Dr. Jerry Turner always laughs loud and he talks fast. He's full of energy. He's got great ideas, a truly passionate scientist. He also likes to take photos of the people in his group when they're doing bench work, and he posts them on his website. And She said the lab was very dynamic, really interesting and intelligent people from a range of different cultures. So she's very grateful. She also said he was a generous mentor. One year, he invited lab members who didn't have family in the States to his home for Thanksgiving. And it was her first Thanksgiving dinner in the United States, and she has great memories. The food was delicious. The house was warm. It was winter in Chicago, so it was cold, yet it was beautiful. So she was very grateful for that. She does have one piece of advice for junior colleagues. PayWing encourages you to explore different curricula. Never stop thinking, always have fun exploring. You'll find interesting things and surprises when you do. And in that spirit, I would like to pause for a moment and send best wishes to any of our listeners who are soon setting off for college or for graduate school this fall. And I'd like to give a special shout out to one of our listeners, Drew Dunham. He's an avid fan of the podcast, and he'll be moving from Ann Arbor to Berkeley to start graduate work in microbiology.
Nice.
So good luck, Drew.
Hope his car makes it all the way across the Rockies.
Bingkong Zhang was a visiting professor in the lab, and he contributed to this work. He was born in the Hunan province in China. He got his bachelor's degree in animal science at the College of Animal Science and Technology at the Hunan Agricultural University there. He then got both a master's and a PhD in animal nutrition at the College of Animal Science and Technology at the China Agricultural University in Beijing. And he worked with Professor Gao studying effects of zinc chloride in the diets of weanling piglets and mechanisms thereof. Since 2008, he's been an associate professor at that same institution and department. And there, his lab focuses on the gastrointestinal tract. And he recognizes that this is an interface with the outside world that's absolutely essential for health. Even modest perturbations could lead to diarrhea, malnutrition, infectious disease, intestinal inflammatory diseases, etc. So they want to identify key microRNAs that regulate and modulate the integrity of epithelial tight junction permeability as a way to promote health in animals. They're also interested in exploring how gut microbes can improve muscle and therefore meat quality. And they've actually had some success with Clostridium butyricium as a probiotic to improve meat quality of broiler chickens. And they found that this Clostridium species can enhance fatty acid uptake and increase intramuscular fat content and therefore improve the meat quality and fatty acid profiles of the breast meat in male broilers. So they're also looking at the effects of this probiotic on the aroma compounds made by the chickens. And in his free time, he enjoys basketball, badminton, and mountain climbing.
Neat.
And what was the last?
Mountain climbing.
Mountain climbing. Oh, nice.
You don't have those in San Diego, right?
Yes, we do.
There's mountains in San Diego.
Yeah, they're little hills.
We have a 6,000-foot mountain within city limits.
Really?
Wow.
Yes, sir.
Wow, that's not a Rocky Mountain, though.
And in the county, there's 8,000.
Oh, my apologies to San Diego.
And get letters.
We're very sensitive.
I have to make fun because you have such nice weather that we have to make fun of something else, right? Thank you, Michele.
My pleasure. I want to thank Jerry Turner for putting me in touch with the two scientists who led the project.
Do you know him? Did you know him before?
I don't.
No? Now you do. All right, a couple of emails before we wrap it up. The first is from Gavin. Dear TWiM, in episode 157 on moon milk, there was some discussion about its medicinal properties. Calcium carbonate is the active ingredient in Tums. Did you know that?
Yeah, when I was listening to you guys, I was sort of saying, "Where's the Tums?" I was screaming at the.
Well, thanks for pointing it out.
In addition, Elio might find it interesting that calcium carbonate is a very popular additive in the cultivation of fungi. Amateur mushroom growers often add ground-up oyster shells to improve growth.
I guess I knew that.
You knew that? Yeah. So Gavin is a medical student at University of California, San Francisco, the class of 2021. Thank you, Gavin.
Very nice. Lovely.
Then we have an email from Neil Greenspan, who is a professor at Case Western. He's writing to Vincent and Arturo. I listened with great interest to TWiM 154 with Arturo, a longtime friend. Clearly, Arturo is both one of the most thoughtful individuals in biomedical science and one of the most prolific. While I have a high regard for Arturo's intellect and views, I persist in formulating my own perspectives on many of the topics he addresses. Frequently, I agree with Arturo's take, but sometimes my view diverges. So I have several comments regarding the conversation of TWiM 154. And just to remind listeners, that was the one we did at ASM Microbe in New Orleans.
Alive.
Pathogenic potential. Overall, Arturo's concept of pathogenic potential is an advance because it highlights the limitations of current notions of virulence. I completely support his critique of the usual classification scheme. As Arturo may recall, I have devoted a fair amount of time and effort to writing about these limitations. With respect to the term pathogenic potential, I am not certain it is the best possible choice. Alternatives I would suggest are specific pathogenic activity or pathogenic potency, with explicit recognition that the cited activity or potency is for the endpoint measured and may not be generally true for all endpoints. In various branches of chemistry, specific activity or potency refers to the magnitude of an effect per unit concentration, and essentially Arturo's ratio would seem to be an analogous variable for microbes. As noted above, an important issue is how the ratio being determined will vary for different endpoints, mortality, weight loss, etc. I would not be surprised to find examples of one pathogen or pathogen strain being more potent for endpoint 1 and less potent for endpoint 2 in comparison to another pathogen or strain in a given host. Similar issues could arise for different hosts with a single pathogen. Measurement of pathogenic potential is worth trying, but I wonder how quantitatively similar assessments of a particular pathogen strain will be for the same inbred mouse strain in different locations. Here again, I would not be surprised by significant differences due to either cryptic genetic differences or variation in relevant environmental factors.
Like the microbiome.
Exactly. Also, there are other limitations of the standard concepts of virulence and virulence factors not directly addressed by the pathogen potential. For example, as I have plans to write about, there is now increasing evidence that optimal virulence is a property of a microbial population, not individual microbes. In addition, there is some evidence consistent with significant interactions between different microbes, such that, for example, a pathogen can perhaps indirectly transform a putatively non-pathogenic microbe into a mediator of host tissue damage. Another example of an interaction for which there is some evidence would be putatively non-pathogenic microbes influencing the ability of pathogenic microbes to infect. Okay, scientific rigor. I agree with Arturo and Farrokh Fang regarding the 5 elements of rigor that they outline. They're all necessary, but I'm not certain if they are all that is needed, or at least all that needs to be brought to the attention of investigators. In addition to being concerned about error, investigators must also be concerned about artifacts, i.e. results which may reveal some aspects of the assay system used, but not reveal much about the underlying biological phenomenon. And he gives an example of that using green fluorescent protein. With respect to logic, the classical logical fallacies are not the sole problems. Failure to use appropriate conceptualizations of phenomena or recognize the limitations of any one scheme can also lead to serious errors of influence. Arturo and Ferrick are correct that logic is not generally taught to graduate students as a formal part of their coursework, but they fall prey to the central limitation of inductive reasoning by claiming that, with the exception of statistics, none of these disciplines are formally taught in the training of scientists. And he gives an example of how he's done this over the years. And finally, a funding lottery. I agree with many of the points. However, I want to note that others, such as my former case colleague David Kaplan, previously pointed out the underpowered nature of peer review at study sections. And he also have mentioned the concept of a lottery. I want to make one other point about ranking grants based on a standard NIH score. Grants like pathogens with respect to virulence have many dimensions, i.e. they are more effectively represented as vectors in a space with 2 or more orthogonal axes than as points along a single line. Anytime multidimensional vectors are reduced to scalars, information is lost. Rankings necessarily require scores that can be ordered, and scores for multidimensional entities necessarily require applying weights to the different variables corresponding to the multiple axes. I'm a little less enthusiastic about moonshot science. The Genome Project was a success in certain respects, but it was sold to Congress and the public somewhat dishonestly as the best path forward to achieve disease cures, a manifestly overstated claim. As Joseph Goldstein, eminent geneticist and Nobel laureate, wrote many years ago, a gene is not a drug. Also, big science projects tend to concentrate authority and power in fewer hands and fewer brains, and by the nature of the relevant selection process, these individuals sometimes appear to be above average in the desire for self-promotion and self-aggrandizement.
Oops. Do you think so?
Furthermore, the projects not pursued or otherwise damaged due to new focus cannot be measured, so an honest and fair assessment of the net worth of the Moonshot Project is extremely challenging, if not impossible. Thus, big science is likely to generate BS. Best regards, Neil. And Arturo actually wrote to him and us and thanked him for the critique. And he said he basically agrees. I have no essential disagreement with any of the points you raise. And I have a suggestion. Why not take the points and write letters to the editor? Of course, Arturo, of course, is the editor-in-chief of MBio. So perhaps Neil will write these up. However, I would say that he will have a bigger audience in TWiM than writing a letter to the editor of MBio. And it will be a more diverse audience.
That's for sure.
Well, and they can be synergistic. We can do both.
Well, he can do both for sure. Absolutely.
Mm-hmm.
But I think this is the power of the podcast, that this is very cool. Anyway, so I will stop there and we will read our other emails next time. That is TWiM 158. You can find it at Apple Podcasts. You can find it at asm.org/TWiM. You should subscribe if you have a smartphone or a tablet. You know, there are apps that you can use to listen to podcasts. Many people do that. Why don't you subscribe so you automatically get them when they are released? You don't have to go looking for them. And it also helps us. The number of subscribers we have impact our ability to raise money by advertising. And speaking of raising money, consider helping us. If you like what we do, give us some financial help. You can go to microbe.tv/contribute, where we have a variety of ways where you can help us. You can open a Patreon account where you could contribute $1 a month, or there are several other mechanisms as well. And we love getting your questions and comments. Send them to twim@microbe.tv. Wow, the whole TWiM team was here. This is unusual for August. Someone is usually away. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you.
Elio Schaechter can be found at lovely blog, Small Things Considered. Thank you, Elio.
Well, thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks to everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society of Microbiology for their support of TWiM, and Chris Condian and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.